Bismillahirrahmanirrahim. Welcome to my YouTube channel Ishaq Tutorial. Please share, subscribe and watch. Thank you. Today's topic of presentation is polymerase chain reaction, or simply it is called PCR. Under this topic we will discuss about the definition of polymerase chain reaction, the invention of polymerase chain reaction, PCR machine or thermocycler, mechanism of polymerase chain reaction, as well as requirement of polymerase chain reaction and uses and application of polymerase chain reaction. As you know that polymerase chain reaction is discovered by an American scientist, Carey B. Mullis, in 1985. This scientist awarded Nobel Prize in 1993. Polymerase chain reaction can be defined that it is an in vitro process that amplifies or copies a piece of DNA repeatedly until there is an amount which is great enough to observe visually. Or it can also be defined in another way, and these techniques or in this process or procedure, DNA polymerase enzyme is compelled to polymerase a given piece of DNA again and again, so that multiple copies are produced. This technique is known as polymerase chain reaction. इस तरीके में हम एक डिजायर्ड जीन को लेते हैं और इसकी डीएनए पॉलीमरेज एंजाइम के जरिए से थर्मोसाइक्लर मशीन में इसकी बहुत ज़्यादा काफियाँ या मौजूदा मिकदार तक काफियाँ बनाते हैं. There is a misconception between polymerase chain reaction and thermocycler. These both terms are often misunderstood. Polymerase chain reaction is basically a procedure. It is a technique. It is a process. While thermocycler is the machine in which the chain reaction is carried out. In thermocycler machine, basically it is a temperature regulator machine. It regulates the temperature. Now we'll go to discuss about the components or requirement of polymerase chain reaction. The first one is template DNA. Template DNA is also known as target DNA. It is a piece of DNA to be copied or to be cloned. Second one is deoxyribonucleotide triphosphate. These are the prenucleotide which is used as a raw material for the synthesis of new DNA fragments. It has 4 different types: deoxyadenosine triphosphate, deoxyguanosine triphosphate, and deoxycytosine triphosphate, and deoxythymidine triphosphate. The third one is primers, a short segments of oligonucleotide of RNA which is used to attach the template DNA. It has 2 types: forward primers, reverse primers, or backward primers. And the fourth one is thermus aquaticus take polymerase. This it is a temperature tolerant enzyme isolated from a bacterium known as thermus aquaticus. This enzyme is stable and active at near boiling temperatures. These 4 components, template DNA, DNTP, 4 different types: primers, forward as well as reverse, and take polymerase. They are mixed with one another, and the mixture is known as reaction mixture or polymerase chain reaction mixture. Now we will go to the mechanism of polymerase chain reaction, which is also called steps of polymerase chain reaction. The first step is denaturation, the conversion of double-stranded DNA into single-stranded DNA. This process takes place at 94 degrees centigrade under a machine known as PCR machine, in which the bonds between the 2 DNA strands is broken down and the double-stranded DNA is converted into single-stranded DNA. It takes 5 minutes for initial cycle, while for repeated cycle it takes only 30 seconds. The second step is known as primer annealing, the attachment of forward as well as backwards or reverse primers at temperature 55 degrees centigrade. It takes 30-second time, in which the forward as well as the backward primers will attach to the template DNA at their required temperatures. So this process is known as primer annealing or simply annealing. You see in the video very clearly that yellow color primers, forward as well as backwards, are attached to the target DNA or to the template DNA. The third step is elongation. Here, attachment of free nucleotide, raw nucleotide, which is present in the reaction mixture, PCR mixture, they will attach by the thermus aquaticus polymerase, DNA polymerase enzyme at 72 degrees centigrade. It requires 30 seconds per cycle, and the total procedure or process completed and it takes 7 to 8 minute times. Now you see in the video that how a double-stranded DNA is converted into single-stranded DNA, then how primers are attached, and how a single-stranded DNA is converted into double-stranded DNA by using raw materials. After the completion of one polymerase chain reaction, a single DNA is converted into 2 copies of DNA. There is an activity if a gene has been amplified 2 times after cycle 1, 4 times after cycle 2, and 8 times in cycle 3 of PCR, how many times the genes will be amplified after cycle 5? So 32 DNA copies are produced. How many? 32 DNA copies are produced. Now we'll go to the uses or application of polymerase chain reaction. It is used for the diagnosis test for specific infection against hepatitis C virus, hepatitis B virus, and hepatitis A virus, as well as for human immunovirus or AIDS virus. For the detection of microorganisms in food, water, or in environment, either in the atmosphere, etc., etc. It is also used for DNA fingerprinting or DNA profiling for identification of criminals. The polymerase chain reaction is also used for solving fraternity cases. Any question? Write your question in the comment box below. I will try my best. Thank you.